Welcome back, everybody. This is another video of biology of cells and tissues. Today we're going to be talking about digestive glands. This is going to be a nicely paired lecture with the digestive system. I'm recording the glands one first. It'll kind of give a good amount of subtext, and then we'll dive into the actual full digestive system and talk about the whole process of digestion. So we're going to start with at the very beginning of the digestive system, which is in the mouth with the salivary glands. We're going to talk about three of them: the parotid, submandibular, and sublingual glands. They're going to produce saliva, which has several different functions, and then we'll also talk about their innervation. So salivary glands produce saliva. The purpose of them, loop of saliva, is to lubricate or moisten food. Starting digestion, it has a specific enzyme, the enzyme amylase, which breaks down starches, which is kind of our carbohydrates, as well as it has an immune function with IgA. Remember, IG stands for immunoglobulin A, as well as another enzyme called lysozyme. This is an antibacterial, it has antibacterial properties, and it breaks down the cell wall of bacteria. One other thing, just noting some vocab that's going to be popping up here and there. Bolus, right here, food turns into a bolus. So if you've ever taken a big bite, if you've ever done something like the saltine challenge, for example, this is a really good example where you have to eat so many saltines or so many crackers ever in a minute, and all of the food kind of turns into the one big blob. That's what we refer to as a bolus, okay? So when you consume food and you chew it all up and it turns into a little blob, and then you're kind of naturally it gets pushed to the back of your throat and you swallow it. As soon as you start to swallow, that is what is known as the bolus. It's going to go down your esophagus and then go into your stomach, and we'll see another word that's called chyme. Chyme is the food and all of your digestive juices all mixed together. We're going to see those words again as we go through a lot of our digestive system stuff. When we talk about innervation, there are two. It's innervated by the autonomic nervous system, which we know has two different branches. It has the parasympathetic, and then it has the sympathetic nervous system. Parasympathetic is our rest, digest. Sympathetic is obviously our fight or flight. So with the parasympathetic nervous system, this is where it produces a large amount of watery saliva that has very little protein in it. This is kind of like when you smell, let's say you smell a familiar food, or you smell dinner that's being prepared, and your mouth starts to water, and there's a lot of saliva that comes out. That is our parasympathetic nervous system activating. Then our sympathetic nervous system, instead of being large amount of watery, we actually just have a small amount of a thicker or viscous saliva. This is produced in response to stress or during periods of excitement. This picture right here, this is more the flow of saliva as well as some of the anatomy. Let's say anatomy. I will stick with anatomy because we're going to cover flow in a little bit. So just like a lot of our other organs, we have the capsule of connective tissue. So we have a connective tissue capsule, and similarly to many of our other organs, like when we talked about the thymus, how it was divided into lobes and lobules, same thing here. A lot of these glands are divided into lobules. It's just kind of the way that they're put together. We're going to see a couple of different types of cells. Serous acini is a type of cell, mucus acini, and then myoepithelial cells. Remember that root word myo. Myo refers to muscle, so these are going to be muscular epithelial cells, okay? As well, the acini, these are clusters of cells that are going to be producing our saliva. We have serous acini, so the serous is the watery, and then mucus, which is more that thicker mucousy saliva. Myoepithelial cells, because they're muscles, they are contractile cells. And then we'll also talk about ducts. The ducts, in particular, intercalated, striated, excretory, and the main duct. We'll see those again in a second and talk about those. So here is just a picture of that salivary gland, some of these different cells. These are the serous, acini, the mucus tubule made up of the mucus, acini, some of these ducts going through here. We'll talk about those in a minute. When we talk about ducts, we've seen pictures similar to this before. When we talked about glands previously, we talked about our endocrine or exocrine glands. Now we talk about more of the compound ducts. So this duct right here is what represents our salivary gland, these compound tubulo-alveolar. These can also be called tubulo-acinar. It means the exact same thing. If you see either of those, that terminology, understand it means the same thing. And when you break it down, compound is just saying it has multiple branches rather than just one singular one. And then the tubulo-alveolar part describes its shape. It has tubular, it has the tube-likes, and then alveolar, again, aka acinar, is like rounded or sac-like structures. Okay, so we can see here this portion. This is more tubular right in through here versus acinar right down there. Okay, so tubular and acinar. So this is the one that we want to make sure we focus on specifically for this chapter. So, talking about the process of saliva secretion. So saliva is produced and secreted by the acini, either the serous or the mucus, into what is called the acinus. The acinus is a small sac-like cavity surrounded by the secretory cells, so the cells secreting. So just like here, little mucus tubule, or you could even say like these little sac-like portions. I think there's a better word for it. These little cups, I guess you could call them. These little, like inside of a little bit of balloon. Those are our acinus. Okay, so the acini is secreted into that area, and then the myoepithelial cells surrounds that whole area. So if we were to zoom way deeper into all of this portion, you'd have those myoepithelial cells around it, just like this, around the edge. And those cells, once saliva has been produced, they're going to contract and they're going to squeeze the saliva along. So it gets squeezed out of the acinus by those myoepithelial cells, and then it enters the intercalated ducts. So produced in the acini cells, goes into the acinus. The myoepithelial cells squeeze and push it into the intercalated ducts. From there, it continues into the striated duct, and then continues into a larger excretory duct right in here, and then it continues, and later on it'll continue and it goes into the main duct, where it would then exit into, in this case, the oral cavity. So what we really want to remember about each of these different ducts specifically is found in this slide. Questions can come from this specific portion, being what type of epithelial cells are we looking at that surround this? So we have simple cuboidal, we have simple columnar, stratified cuboidal, and then stratified squamous. So make sure that you pop those into a Quizlet or something so you understand what type of epithelial tissue is connected to each portion of those ducts. All right? Now, those are kind of the functions and saliva secretions. Now we're going to get into the glands themselves. Here's just a little bit of anatomy, kind of right in here, showing where these glands are located. Parotid glands right up here. We have our submandibular glands. Think submandible below the mandible, and then sublingual below the tongue. So first off, the parotid gland. The main things that we're going to want to remember about these glands specifically is the type of ducts. I would say the type of compound duct that they are made up of, or that is most commonly seen in these, as well as some of the other little tidbits. So, for example, the parotid gland, good little fact to know, it is the largest salivary gland. It sits right over that, also the largest muscle of mastication being the masseter. I know in DAA, or in anatomy courses, you guys have talked about muscles of mastication. And then we have, it is a compound acinar gland. So no tubules, tubes, whatever the proper anatomical word is. There's no tubulo part of this gland. It's just acinar gland, which means it just has those rounded sacs. And it produces a lot of our serous, or the watery saliva, which means the one that has lots of amylase. Remember, what does amylase break down? It starts to break down starches and carbohydrates in our mouth. And then it also has plasma cells in the connective tissue, as well as IgA. Here is our parotid gland, kind of what it looks like. We have those serous cells. Let me make this picture just a little bit bigger. Lots and lots of serous cells. Next, our submandibular gland. So even though the parotid gland is the largest, the submandibular gland produces most of the saliva. So the submandibular gland produces 2/3 of all saliva. This one is a compound tubuloacinar gland. It has the tubes and it has the sacs. And this has both the mucus and the serous acini cells, as well as serous demilunes. I believe I'm pronouncing that correctly. Serous demilunes. These are these crescent-shaped cells that cap the ends of the mucus acini. And these secrete those lysozymes. Remember, lysozymes are for antibacterial properties. Here's our submandibular gland. These cells kind of surrounding on the outside. You have our mucus cells, our mucus acini cells, and these are our serous demilunes. So thinking kind of more on the lab side of things, when you go to take the lab exam, I would look specifically for what kind of things make this different. Notice in the parotid gland, we only have serous cells, so lots and lots of serous cells. In the submandibular gland, we have the mucus cells, which seem much more square-esque, as well as these demilunes. And then finally, the sublingual gland is also tubuloacinar. Remember, tubuloacinar, tubuloalveolar, it means the same thing. So those words are interchangeable with each other, okay? I guess I should clarify. They are, for the most part, used interchangeably. They do technically, like if you get into like the nitty-gritty of them, they do technically mean different things. However, in most situations and in this course, they're used interchangeably, so don't worry too much about that. Again, it has both serous and mucus acini cells, but in this case, the serous cells have the demilunes instead of the mucus cells like the I apologize. It's both the same. These also have the demilunes on the serous cells. Little clinical stuff. A couple words. Dry mouth, also known as xerostomia. You're going to start to see these kind of words pop up with this kind of X's and funky spellings. These are type of words that board exams, part 1 especially, really, really likes. So start kind of compiling a list, keeping these in the back of your mind, and understand that, hey, these are going to come back around, and you need to know these words for board exams in the future. It can be associated, so dry mouth can be associated with anything that affects the salivary glands. like the mumps, viral infection, radiation, or normal side effects of drugs such as antihistamines. So allergy medication. Okay, finished with the lingual, or should I say the salivary glands, we're going to move on to the pancreas. So the pancreas is never going away. You're going to talk so much about the pancreas in so many of the courses because it is an incredibly important organ, and we'll dive into why. So of course, just like all the ones we've seen, it has a capsule with thin connective tissue. You have what are called septa, which is just a continuation of the capsule that just dives deeper and it divides it into lobules. It's a common theme here. And then we have an exocrine and an endocrine portion of these glands. These are very, very important. The endocrine portion is known as the isolates of Langerhans, and it makes up about 1 to 2% of the total gland. So overall of the entire gland, it seems relatively small, but it plays a very, very important role. And then we have the exocrine gland, which is a compound acinar gland. Again, we'll want to remember the type of gland for potential test questions. Here's what we see. We see the pancreas, and the pancreas is attached right to the duodenum. Duodenum, duodenum, however you want to pronounce it. Anatomy is weird where they don't have a set pronunciation of a lot of these words. But if we know that the duodenum, specifically right here, this is the duodenum here, and this is actually connected right to the stomach right here. Okay, so the duodenum is the very first part of the intestines, where remember what that word that I used, that chyme goes from the stomach and enters into the small intestine. Again, here we have all these exocrine portion. The lighter portion are our endocrine portions, these isolates of Langerhans. Everywhere else, all this other stuff is exocrine. And real quick, reminder that exocrine versus endocrine, we're referring to where the things that they produce go. Exocrine means that they produce stuff, enzymes or whatever, or what have you, that are eventually going to actually exit the body. It's like sweat glands are a most common example of exocrine. Endocrine means that it's whatever it's producing stays inside of the body. Okay? So these isolates of Langerhans, there are 4 types of cells: alpha cells, beta cells, delta cells. These are the big 3 that we always talk about. But then there's also an extra 4th called PP cells, or also known as F cells. Okay? We want to associate each of these cells with what they produce and their function. So alpha cells produce the hormone glucagon, and glucagon regulates the blood sugar by increasing blood glucose levels when needed. You're going to cover this in other courses, but I'm just going to throw it in here. Glucagon acts primarily on the liver, as the liver stores a bunch of the metabolites, fats, glucose, that sort of stuff that we need. And so most of the glucagon is going to act on the liver in order to raise the blood glucose levels when we have not eaten food or we don't have enough blood glucose in our bodies. And you'll talk more about this a lot in like biochemistry as well in a handful of other courses. So, alpha cells, glucagon to increase blood glucose. Beta cells produce insulin, and insulin decreases blood glucose levels. And what it does is it causes the cells of the body to uptake the glucose from the blood. Delta cells, these types of cells produce a hormone known as somatostatin, and this hormone actually inhibits glucagon and insulin. Somatostatin, you'll learn more about somatostatin in Physiology 2 and Trimester 3. But you need to know that it comes from the delta cells. PP or F cells, these produce the hormone pancreatic polypeptide. And this blocks the release of another hormone known as cholecystokinin, which we're going to talk about a little bit after this. We'll talk about the specific hormone a little bit more later, a little bit more in the future. So that's the endocrine portion. Remember those isolates of Langerhans. And now we're going to talk about the exocrine portion. Most of it has to do with digestion. Okay? Digestive enzymes. So we have these cells called centroacinar cells, which secrete bicarbonate in order to neutralize the enzymatic secretion of the acini. There's more to it there, but I want to make sure in Physiology 2, you dive deeper into all of this again. So I'm going to leave it at that for now. You have another duct. So I'll go over the process of the ducts, kind of the order of them. But then you also have the main pancreatic excretory duct, also known as the duct of Wirsung. And then you have an accessory duct known as the duct of Santorini. I included a bunch of extra information down in here, so we'll just kind of overall summary. If you want to dive deeper a little bit and read about that, go right ahead. But it's not super important at this time, all this information. So here's the main duct along the center of the pancreas. So again, all of these exocrine cells all over here are producing enzymes and then putting them into the main duct, and then they come down in here, and they can either go through the accessory duct or go directly through, and it meets up actually with one of the ducts of the gallbladder, which we'll mention, and then goes into the small intestine. Here's what the exocrine pancreas looks like. We have these centroacinar cells, an intercalated duct, which leads to that main duct, as well as acinar cells and a couple of other things. Here are some of the other secretions that they produce. Chemicals or enzymes such as trypsinogen, carboxypeptidase, and chymotrypsinogen. These are responsible for breaking down different types of proteins. Amylase breaks down carbs, lipase breaks down fats, and then nuclease breaks down nucleic acids. Commit these to memory, as they will come back many times. Here's that word we're going to talk about, chyme. So chyme that leaves the stomach and goes into the small intestine, into the duodenum, stimulates these cells that are known as enteroendocrine cells to release two hormones, secretin and cholecystokinin, which we mentioned cholecystokinin before. Secretin acts on the cells of the pancreas and promotes water and bicarbonate secretion by the duct cells. It produces bicarbonate, again, because chyme coming from the stomach is acidic, and we need a way to neutralize that, so that way our intestines don't get burned by that low pH chyme. Cholecystokinin is another hormone that stimulates digestive enzymes that are secreted by the acinar cells, so it stimulates the release of pancreatic digestive enzymes. So what we really need to know is the specific hormones and what they do. So secretin is paired with bicarbonate release, cholecystokinin with the release of pancreatic digestive enzymes, such as all of these guys. Just a picture of kind of this whole process as a whole. You don't need to worry too much about this specific picture. On to the liver. So the liver, there's a whole bunch of parts of the liver. Really, what you want to remember with the liver, anatomy-wise, is more for like DAA and for other courses. But we want to know the functions and and what the liver does really well here. So the liver has endocrine, exocrine, metabolic storage, and urea production functions. Its endocrine function, it produces plasma proteins like albumin, globulins, etc. Albumin and globulins are super important for maintaining blood pressure. Okay, liver plays a very important role in maintaining our blood pressure by producing those plasma proteins. It also produces a substance called bile. Bile is a fluid produced in the liver, then stored in the gallbladder. And when it goes into the intestines, its job is to emulsify fats. Kind of what that means. Emulsification is kind of like mixing, where it mixes with the fat and kind of like teases and separates this fat molecules away from each other to allow for enzymes to come in and do their thing in order for the lipase enzyme to break down the fat. Because fats, oftentimes they like to kind of clump together, so especially in like the mixture that they're in the stomach. So we need a way to kind of separate and tease them away from each other in order to be able to digest them properly. And that is what bile does. You'll talk about bile and bile salts in biochemistry as well. So you'll see quite a bit of that. And as well, bile is going to come back a lot when we talk about bile, and the gallbladder are going to come back a lot when we talk about pathologies and stuff in the future. Metabolite storage. The liver stores important stuff. Lipids, so fats, as triglycerides, carbohydrates as glycogen, and then fat-soluble vitamins, especially vitamin A, but also vitamins D, E, and K. Way to remember this, I've mentioned this before in biochemistry. A, fat, dec, is the way that we can remember all of our fat-soluble vitamins. Finally, the liver's another function of the liver is urea production. So the one thing in our body that we really can't store is our proteins and amino acids. We produce them, we use them, and then that's kind of it, okay? So when proteins are no longer useful to us, we break them down, and they need a place to go. So what happens is when they get broken down, the nitrogen part of amino acids, remember amino part is the nitrogen that's on there, the NH3. So that nitrogen is removed in a process called deamination, but because my nitrogen is toxic, the liver then converts it to urea. You'll learn this whole process in biochemistry. And then that urea can be filtered in the kidneys and removed via the urine. Urine, urea, that's kind of why it's named the way that it is. So like I said, you'll talk more about this in biochemistry. Again, liver, talk about the stroma. We should know that word by now. Remember stroma, kind of meaning skeleton, meaning like those supporting type cells. There's a thin connective tissue capsule. There's a hilum, kind of like what we talked about with the nodes, with the lymph nodes. Remember, where the hilum is where stuff enters and exits. You have portal veins and a hepatic artery enter there, and hepatic ducts and lymphatics exit. And the stroma is made up of reticular connective tissue. Getting a little bit more specific, we talk about, again, it's divided from like the whole liver. There are lobes of the liver, and then we look even deeper. We have these things called lobules. There is a central vein in the center of the lobule, and then there are portal areas at the periphery, or the outside of the lobules. So this right here is a lobule. All those little dots are hepatocytes, or liver cells. So we have our lobule right there. It's kind of hexagon shape. And then we have on the outside of them, or on the periphery, we have that bile duct, hepatic artery, and the portal vein. And then we have the central vein in the middle of the lobule. This is all really good testable material, as well as lab, especially lab. You'll get questions, quite a few questions on the liver with pictures and stuff like this. More of the lobule, central vein, hepatocytes, all of this kind of stuff. If you see three things together, understand and know that that is the portal areas. Just another picture of all of this. We'll talk about bile ducts in a sec. So we have the blood supply of the liver. The liver has what's called dual blood supply, which means it receives blood both from the venous side as well as the arterial side. 80% of its blood comes from the venous side, from the hepatic portal vein. And this is nutrient-rich blood because it's coming right from the intestinal tract. So we get 80%, we get a lot of nutrient-rich blood in order to, do and store and do whatever we need to do with whatever's in the blood. And then the artery side, which is 20%, we get our oxygen in order to feed the actual tissues that oxygen that we need. Much like in the spleen, we talk about sinusoids. The liver also contains specialized large capillary sinusoids. Okay? Very, very similar to what we saw with the spleen in the previous, in the lymph lecture, cleaning the blood, that kind of stuff. So there's that discontinuous fenestrated endothelial cells, which have these specialized macrophages called Kupffer cells. So we really just want to make the connection. Liver has Kupffer cells. Kupffer cells are the immune cells of the liver. There's another piece of anatomy called the space of Dicey. This is just an area between the endothelial cells and the liver cells. So here's the endothelial cells and the hepatocytes right there. And this is where a lot of our exchange happens, like nutrient exchange and waste exchange, that kind of stuff. Talking a little bit more about the hepatocytes. We also call, it's a little bit confusing, we call both the hepatocytes and the lobule the functional units of the liver, because essentially the lobules are made up of hepatocytes, so like they're kind of grouped together. The lobules are made up of kind of these plates of cells. We have a polyhedral shape. The apical, or the top surface, is open to the sinusoids. The basolateral surface is attached to other hepatocytes. A lot of this is just kind of random info. Biggest thing I'd say is know the shape, polyhedral shape. We mentioned a couple times about bile and bile ducts. So this is the flow chart of bile production. Remember, bile, what is it? It's a fluid that it's secreted. It's produced by the liver, stored in the gallbladder, and then released into the intestine in order to help us digest fats. It breaks apart the fat so that the lipase released by the pancreas can break it all down. So here's the process. The hepatocytes produce the bile and ducts drain it away. We have what are called bile canaliculi or canaliculi when it's singular. These are small channels between the hepatocytes that collect bile. We have these drain together into a bile ductule, and then those drain together into the bile ducts of the portal triads. Then it continues on its path, converging, getting bigger. Then you have right and left hepatic ducts. They come together to form the common hepatic duct, and then the cystic duct, and then the common bile duct. It's just a path down, so you get these little canaliculi. I thought there was a better picture. Let me pull up a picture. Here we go. Here's a little bit easier picture to see. So we have those right and left hepatic ducts coming together into the common hepatic duct. And then we have a cystic duct, which leads to the gallbladder, and then the common bile duct, which goes down, and we saw actually a part of that looking at the pancreas. There's the common bile duct leading into the same spot as our main duct. Which brings us actually to this little organ called the gallbladder. For some reason, a lot of people have trouble with gallbladder specific stuff, so I want to make sure you see this word right here, cholecyst. Okay, anytime you see this word cholecyst at the beginning, so you'll see cholecystitis, you'll see cholelithiasis, nephrolithiasis, I think is the pronunciation of it. You'll see a lot of these words, cholecyst. If that's at the beginning, understand you're talking about the gallbladder, okay? The function of the gallbladder is the bile that is produced in the liver comes down, goes through the common hepatic duct, and then comes up through the cystic duct and is stored in the gallbladder. As it is stored in the gallbladder actually removes water from it in order to concentrate the bile. It has a mucosal layer, which is made up of simple columnar epithelial, and connective tissue binds it to the surface of the liver. If it's attached directly to the liver, so like these portions here, connected right on the liver, that's like right up against the liver, we call that adventitia. This is just kind of vocab stuff. And when it is on a free surface, so like this part down here, where it's not in contact with the liver, we call that serosa. Just because, I guess, anatomists wanted to make it complicated. Not entirely true. There's a reason why it's said different, but we don't need to dig that deep. There's the gallbladder, the hepatic duct, the cystic duct, and then the common duct. And these actually come together right here. And there's the main pancreatic duct, and these run together to go into where? Into the duodenum of the small intestine. And here is, you can see the tissue of it. You have the adventitia, the muscularis, fluid-filled mucosa, and this is the actual gallbladder itself. So yeah, and that is all of our digestive glands. Next video, we're going to talk about the digestive system kind of as a whole. So yeah, getting through it. As always, if you have any questions, comments, concerns, drop them down below and I'll get to them. Leave a like on the video if you can, subscribe if you haven't already, and I will see you on the next one. Have a great day and good luck studying.